Hello, this is Dr. Morris. I am doing lecture number 3, concerning the chemical and molecular human anatomy. The lecture today will start with the electrolytes and the body fluids that we see in the human system. Soluble inorganic molecules are typically those whose are ions will conduct an electrical current in a solution called electrolytes. Sodium chloride is an electrolyte, and sodium chloride is one of the major electrolytes, and we'll discuss it a lot more in detail later on in subsequent lectures dealing with the human body. The dissociation of electrolytes in blood and other body fluids releases sodium ions, potassium ions, calcium ions, and chloride ions. Concentrations of electrolytes. Changes in the concentration of electrolytes in the body fluids will disturb almost every vital function. For example, declining potassium levels will lead to general muscular paralysis. Rising concentrations will also cause weak and irregular heartbeats. The concentration of ions in body fluids are carefully regulated primarily by coordination of activities at the kidneys, ion excretion for example, the digestive tract with ion absorption, and then in the skeletal system with ion storage and release. Table 2.3 demonstrates important electrolytes that dissociate in body fluids. These include sodium chloride, potassium chloride, calcium chloride, sodium bicarbonate, magnesium chloride, disodium phosphate, and sodium sulfate. Other characteristics that are important in compounds inside the human body include the hydrophilic and hydrophobic compounds. Some organic molecules contain polar covalent bonds which also attract water molecules. The hydration sphere, which we talked about in lecture 2, that form may carry these molecules in solution. Molecules such as glucose is an important soluble sugar that interacts readily with water molecules in this way. It's called hydrophilic. Hydrophilic and hydrophobic compounds. Many of these other organic molecules have few polar covalent bonds or none at all. Such molecules are said to be nonpolar. When nonpolar molecules are composed of water, hydration spheres do not form and the molecules do not dissolve. Molecules that do not readily interact with water are called hydrophobic. Among the most familiar such molecules are fats and oils of all kinds. Solute concentration. The concentration of a substance is the amount of that substance that is present in a specified volume of solvent. Physiologists and clinicians often monitor inorganic and organic solute concentrations in the body fluids such as blood or urine. Solute concentrations. Such solutes has a normal range of values, and variations outside this range may indicate disease. Many solutes are participants in biochemical reactions, and as we noted earlier, the concentrations of reactants and the products in a chemical reaction is directly affect the reaction rate. Colloids and suspensions. Body fluids may contain large and complex organic molecules such as proteins, protein complexes that are held in solution by association with water molecules. A solution containing dispersed proteins or other large molecules is called a colloid. Liquids is a familiar viscous or thick colloid. Suspensions. The particles or molecules in a colloid will remain in a solution indefinitely. A suspension contains an even larger particles that will, if undisturbed, settle out of the solution due to the force of gravity. Stirring beach sand in a bucket of water creates a temporary suspension that will last only until the sand settles to the bottom of the bucket. Whole blood is another temporary suspension. Because the blood cells are suspended in blood plasma, this is why in laboratory testing whole blood solutions can be easily separated from the blood plasma by spinning it. Gravity and centrifugal force will allow the blood cells to settle at the bottom of the suspension, leaving the clear plasma solution. If clotting is prevented, the cells in a blood sample will gradually settle to the bottom of the container, as I described. Measuring that settled rate, sometimes referred to as the sedimentation rate, is a common laboratory study. Hydrogen ions in the body fluid. Hydrogen atoms involved in a chemical bond or participating in chemical reactions can easily lose its electron. To become a hydrogen ions are extremely reactive in solution. In excessive numbers, they will break chemical bonds, change the shapes of complex molecules, and generally disrupt cell and tissue's function. As a result, the concentration of hydrogen ions in a body fluid must be regulated precisely. Hydrogen ion concentration in the body or in body fluids is so important to physiological processes that a special shorthand has been used to express it. The pH of a solution is defined as the negative logarithm of the hydrogen ion concentration in moles per liter. Thus, instead of using an equation, we say the pH of pure water is or 7. Using pH values saves space, but always remember that the pH number is an exponent and that the pH scale is logarithmic. For instance, a pH of 6 really represents 0.000001. This means that the concentration of a hydrogen ion is 10 times as great as it is at a pH of 7. This illustration is taken from your book, demonstrates the pH and hydrogen ion concentration. The scale is a logarithmic scale. An increase or decrease in 1 unit corresponds to a 10-fold change in concentration. This illustration demonstrates some of the common products that we see not only in household products, but also when we look at the human body itself. It demonstrates to you which pH certain environments inside our body may exist. For example, an extreme acidic pH would be hydrochloric acid inside the stomach. As we look to the pH of water, pure water has a neutral pH of 7. Saliva and or milk, which is located either in the mouth, demonstrates a pH somewhere between 6 and 7, meaning that it would be more alkaline than some of the other substances that are in the body as well. As we significantly increase the pH to a more alkaline or more basic solution, you can see some of the household products that represent that. Household bleach, ammonia, oven cleaner being the most extreme alkaline substance noted in this pH scale. pH ranges. Although pure water has a pH of 7, solutions display a wide range of pH values depending on the nature of the solids involved. A solution with a pH of 7 is said to be neutral because it contains an equal number of hydrogen and hydroxide ions. A solution with a pH below 7 is considered to be acidic, meaning that it contains more hydrogen ions than hydroxide ions. A pH of above 7 is basic or alkaline, meaning that it is more hydroxide ions than hydrogen ions. The normal pH range for the human body is between 7.35 and 7.45. You should commit these numbers to memory. Abnormal fluctuation in pH can damage the cells and the tissues by breaking chemical bonds and changing the shapes of proteins and altering cellular functions. Acidosis is an abnormal physiological state caused by low pH, again below the level of 7.35. A pH below 7 can produce a coma. Alkalosis results from an abnormal high pH above 7.45. A blood pH above 7.8 generally causes uncontrollable, sustained skeletal muscle contractions. Inorganic acids and bases. The body contains both inorganic and organic acids and bases that may cause acidosis, alkalosis, respectively. An acid in any solute that dissociates in solution and releases hydrogen ions, thereby lowering the pH. Because a hydrogen atom that loses its electron consists solely of a proton, hydrogen atoms are often referred to simply as protons and acids as proton donors. Inorganic acids and bases. A strong acid dissociates completely in a solution, and the reaction occurs essentially in one direction only. Hydrochloric acid is a representative strong acid in water. It ionizes as the stomach produces this powerful acid to assist in the breakdown of food. Hardware stores sell hydrochloric acid under the same name, muriatic acid, for cleaning sidewalks and swimming pools. A base is a solute that removes hydrogen ions from a solution and thereby acts as a proton acceptor. In a solution, many bases release hydroxide ions. Hydroxide ions have a strong affinity for hydrogen ions and react quickly with them to form water molecules. A strong base dissociates completely in a solution. A strong base, such as sodium hydroxide, is a strong base in solution. It releases sodium ions and hydroxide ions. Strong bases have a variety of industrial and household uses. Drain openers, lye, are two familiar examples of strong bases. Weak acid and weak bases. Weak acid and weak bases fail to dissociate completely. At equilibrium, a significant number of molecules remain intact in solution. For the same number of molecules in solution, a weak acid and weak base therefore have less of an impact on pH than do strong acids and strong bases. Carbonic acid is a weak acid found in body fluids. In a solution, carbonic acid reversibly dissociates into hydrogen ions and bicarbonate ions. A salt is an ionic compound consisting of any cation except a hydrogen ion and any anion except a hydroxide ion. Because they are held together by ionic bonds, many salts dissociate completely in water, releasing cations and anions. Sodium chloride, table salt, dissociates immediately in water, releasing the ions. Sodium and chloride are the most abundant ions in the body fluids. Many other ions are present in lesser amounts. As a result, the dissociation of other inorganic compounds. The ionization of sodium chloride does not affect the local concentration of hydrogen ions or hydroxide ions, so sodium chloride, like many salts, is a neutral solute. Through their interactions with water molecules, however, other salts may indirectly affect concentrations of an ion. Thus, the dissociation of some salts make a solution slightly acidic or slightly basic. Buffers and pH control. Buffers are compounds that stabilize the pH of a solution by removing or replacing hydrogen ions. Buffer systems typically involve a weak acid and its related salts, which function as weak bases. For example, the carbonic acid and bicarbonate buffer system consists of carbonic acid and sodium bicarbonate, otherwise known as baking soda. Buffers and buffer systems in the body system, or the body fluid, maintain the pH within a normal range. Antacids, such as used in sodium bicarbonate to neutralize excess hydrochloric acid in the stomach. The effect of neutralization are most evident when you add a strong acid and a strong base. Adding hydrochloric acid to a sodium hydroxide, you neutralize both the strong acid and the strong base. This reaction produces water and a salt, in this case, the neutral salt, sodium chloride. Organic compounds always contain element carbon and hydrogen, and generally oxygen as well. Many organic molecules are made up of long chains of carbon atoms linked by covalent bonds. The carbon atoms typically form additional covalent bonds with hydrogen or oxygen atoms, and less commonly with nitrogen, phosphorus, sulfur, iron, or other elements. Many organic molecules are soluble in water. There is important organic acids and bases in the body. Lactic acid is an organic acid generated by active muscle tissue that must be neutralized by buffers in the body fluids. There are four major classes of organic compounds. These include carbohydrates, lipids, proteins, and nucleic acids. Another important concept to appreciate are functional groups. Although organic compounds are diverse, certain groupings of atoms occur again and again, even if very different types of molecules are present. These functional groups greatly influence the properties of any molecule that they are a part of. This illustration demonstrates to you the different and various types of functional groups. This is table 2.4 from your textbook. Denotes the functional groups, the structural formulas related to these functional groups, and then the importance of each of these groups, and then some examples by which you would see these functional groups exist. Carbohydrates, the first organic molecule that we'll discuss. A carbohydrate is an organic molecule that contains carbon, hydrogen, oxygen in a ratio of 1 to 2 to 1. Familiar carbohydrates include sugars, starches. They make up roughly half of the typical U.S. diet. Our tissues can break down most carbohydrates, which typically account for less than 1% of the total body weight. Carbohydrates, although they may have other functions, they are most important as a source of energy that are catabolized rather than stored. Students should be familiar with monosaccharides, disaccharides, and polysaccharides, which are various forms of carbohydrates. Monosaccharides, for example, a simple sugar, is a carbohydrate containing 3 to 7 carbon atoms. A monosaccharide can be called a triose, or a 3-carbon, tetrose, 4-carbon, pentose, 5-carbon, hexose, 6-carbon, or heptose, 7-carbon. The hexose, or glucose, is the most important metabolic fuel in the body. Atoms of glucose molecules may form a straight chain or a ring. In the body, the ring form is the most common. A three-dimensional molecule shows the arrangement of atoms in the ring most clearly. This is an example of the glucose molecule. Some molecules have the same molecular formula, in other words, the same types and numbers of atoms, but different structures. Such molecules are called isomers, a term you should become familiar with. The body usually treats different isomers with or as distinct molecules. Fructose is a hexose found in many fruits and in secretions in the male reproductive system, the importance of which will be described when we fully discuss the male and female reproductive systems later in the lecture series. Although its chemical formula is the same as glucose, separate enzymes and reaction sequences control breakdown synthesis. Monosaccharides such as glucose and fructose dissolve readily in water and are rapidly distributed throughout the body, the blood, and other fluids. This is an illustration, again taken from your book, that demonstrates the formation and breakdown of complex sugars. These reactions are performed by enzymes in the cell. The first illustrates a dehydration reaction, second of which demonstrates a hydrolysis reaction. In the dehydration reaction, or dehydration synthesis, two molecules are joined by the removal of a water molecule, the combination of glucose and fructose forming sucrose, and the generation of a water molecule. In B, you see hydrolysis, which reverses the steps of the dehydration synthesis. A complex molecule is broken down by the addition of water. Again, sucrose being broken down into glucose and fructose. As you can see, the water molecule has been separated. Carbohydrates other than simple sugars are complex molecules composed of monosaccharide building blocks. Two monosaccharides joined together form a disaccharide. Disaccharides such as sucrose, table sugar, have a sweet taste and, like monosaccharides, are quite soluble in water. Dehydration synthesis or condensation. The formation of sucrose involves dehydration synthesis. Dehydration synthesis is condensation links molecules together by the removal of water molecules. Hydrolysis. The breakdown of sucrose into simple sugars is an example of hydrolysis, the functional opposite of the dehydration synthesis. Many foods contain disaccharides, but all carbohydrates except monosaccharides must be dissembled through hydrolysis before they can provide any useful energy. Most popular junk foods, such as candy, sodas, abound in monosaccharides, commonly fructose, and disaccharides, generally sucrose. Some people cannot tolerate sugar for medical reasons. Others avoid it in an effort to control their weight. Excess sugars are converted to fats for long-term storage. Artificial sweeteners. Many people use artificial sweeteners in their foods and their beverages. These compounds have a very sweet taste, but they either cannot be broken down in the body or used in significant amounts. Dehydration synthesis can continue adding monosaccharides or combining disaccharides to create increasingly complex carbohydrates. These large molecules are called polysaccharides. Polysaccharide chains can be straight or highly branched. Cellulose. A structural component of many plants is a polysaccharide that our bodies cannot digest. Foods such as celery, which contain cellulose, water, and little else, contribute to the bulk of digestive waste, but are useless as a source of energy. In fact, you expend more energy when you chew a stalk of celery than when you obtain by digesting it. Starches are large polysaccharides formed from glucose molecules. Most starches are manufactured by plants. Our digestive tract can break these molecules into monosaccharides. Starches such as those in potatoes and grains are the major dietary energy sources. Glycogen, a structure that you should become familiar with. The polysaccharide glycogen, or animal starch, has many side branches, all consist of chains of glucose molecules. Like most other starches, glycogen does not dissolve in water or other bodily fluids. Liver tissues and muscle tissues make and store glycogen. When these tissues have a high demand for glucose, glycogen molecules are broken down. When the demand is low, liver and muscle tissue absorb glucose from the bloodstream and rebuild glycogen reserves. Despite their metabolic importance, carbohydrates account for less than 3% of our total body weight. The structure of polysaccharides. Liver and muscle cells store glucose as the polysaccharide glycogen, long branching chains of glucose molecules. This figure uses a different method of representing the carbon ring structure. At each corner of the solid hexagon is a carbon atom. The position of the oxygen atom in each glucose ring is shown in this illustration. Lipids. Like carbohydrates, lipids, also referred to as fats, contain carbon, hydrogen, and oxygen. Lipid contains less oxygen than do carbohydrates, with the same number of carbon atoms. The hydrogen to oxygen ratio is therefore very large. Representative of lipids, such as lauric acid, has a formula which is demonstrated in your textbook. Lipids may also contain small quantities of phosphorus, nitrogen, and sulfur. Familiar lipids include fats, oils, and waxes. Most lipids are soluble in water, but special transport mechanisms carry them in the circulating blood. Lipids form essential structures or components of all cells. Lipid deposits are important as energy reserves. On average, lipids provide roughly twice as much energy as carbohydrates do, gram for gram, when broken down in the body. When the supply of lipids exceeds the demand for energy, the excess is stored in fat deposits. For this reason, there has been a great interest in developing fat substitutes that provide less energy but have the same taste and texture as lipids. Lipids normally account for 12 to 18% of the total body weight of an adult man, and 18 or 24% in the adult female. Many kinds of lipids exist in the body. We will consider 5 classes of lipids: fatty acids, oxycanoids, glycerides, steroids, phospholipids, and glycolipids. Carbohydrates are important as quick source of energy and are integral components in the membranes. Lipids form membranes within and between cells and prevent the diffusion of solids. Fats are large lipids, important as energy reserves. Fatty acids are long carbon chains with hydrogen atoms attached. One end of the carbon chain always bears a carboxylic acid group. The name carboxyl should help you to remember that a carbon and a hydroxyl group, represented by OH, are the important structural features of fatty acids. The carbon chain attached to the carboxylic acid group is known as the hydrocarbon tail of the fatty acid. Representative of fatty acids and lauric acid found in coconut oil and oils of the laurel evergreen. I'm not sure what a laurel evergreen is, but again, it's a source of oil. When a fatty acid is in solution, only the carboxyl end associates with a water molecule, because that is the only hydrophilic portion of the molecule. If you'll go back, you'll remember the definition we gave for hydrophilic and the definition we gave for hydrophobic. Hydrophilic, as it refers to, is the water-loving end, or is in contact with the water medium. The hydrocarbon tail is hydrophobic, so that fatty acids have a very limited solubility in water. Again, hydrophobic typically repels water. In general, the longer the hydrocarbon tail, the lower the solubility of the molecule. When we talk about cellular anatomy in the upcoming lecture series, you will see the importance of the hydrophilic and hydrophobic ends in the cell membrane. Fatty acids may be either saturated or unsaturated. These terms refer to the number of hydrogen atoms bound to the carbon atom in the hydrocarbon tail. Saturated fatty acids. In a saturated fatty acid, each carbon atom in the tail has 4 single covalent bonds. Within the tail, 2 of those bonds, 2 adjacent carbon atoms, and those other 2 bind the hydrogen atoms. The carbon atom at the distal end of the tail binds 3 hydrogen atoms. Unsaturated fatty acids. In an unsaturated fatty acid, 1 or more of a single covalent bond between the carbon atoms has been replaced by a double covalent bond. As a result, the carbon atom involved will each find only 1 hydrogen atom rather than 2. This changes both the shape of the hydrocarbon tail and the way that the fatty acid is metabolized. Monosaturated fatty acids. A monosaturated fatty acid has a single double bond in the hydrocarbon tail. A polyunsaturated fatty acid contains multiple double bonds. And carcinoids are lipids derived from arachidonic acid, a fatty acid that must be absorbed in the diet because it cannot be synthesized by the body. The two major classes of carcinoids are leukotrienes and prostaglandins. Leukotrienes are produced primarily by cells involved in coordinating the response to injury or disease. Prostaglandins are short-chain fatty acids in which 5 of the carbon atoms are joined in a ring. These compounds are released by cells to coordinate or direct local cellular activities and are extremely powerful even in minute quantities. Prostaglandins' effect may vary with the structure and the site where they are released. Prostaglandins released by damaged tissue, for example, stimulate nerve endings producing a sensation of pain. Those released in the uterus help trigger the start of labor contractions. Another category of molecules that you should become familiar with are hormones. The body uses several types of chemical messengers, those that produce in one part of the body but have an effect on a distant part of the body. These are referred to as hormones. Hormones are distributed throughout the body in the bloodstream, whereas most prostaglandins affect only the area in which they are produced. As a result, prostaglandins are often referred to as local hormones. The distinction is not a rigid one. However, as some prostaglandins also enter the bloodstream, they affect other areas to some degree. Glycerides. Unlike monosaccharides, individual fatty acids cannot be strung together in a chain by dehydration synthesis, but they can be attached to one another compound, glycerol, through a similar reaction. The result is a lipid known as glyceride. Dehydration synthesis can produce monoglycerides consisting of glycerol plus one fatty acid. Subsequent reactions can yield a diglyceride, glycerol, 2 fatty acids, and a triglyceride, glycerol, and 3 fatty acids. Excuse me. Hydrolysis breaks the glycerides into fatty acids and glycerol. Dehydration synthesis and hydrolysis operates the same way, whether the molecule involves are carbohydrates or lipids. Triglyceride formation. The formation of triglyceride involves the attachment of fatty acids to a glycerol molecule through dehydration synthesis. A triglyceride may be formed by the attachment of one unsaturated or 2 saturated fatty acids to a glycerol molecule. Triglycerides or triglycerols are neutral fats. 3 important functions. 1. Energy source. Fatty deposits in specialized sites of the body represent a significant energy reserve. In times of need, triglycerides are disassembled by hydrolysis, yielding fatty acids that can be broken down to provide energy. Number 2, insulation. Fat deposits under the skin serve as insulation, slowing heat loss to the environment. Heat loss across a layer of lipids is only about 1/3 that through the tissues. Protection. A fat deposit around a delicate organ such as the kidneys provides cushion and protects it against shocks or blows. Storage of triglycerides. Triglycerides are stored in the body as lipid droplets within cells. The droplets absorb and accumulate lipid-soluble vitamins, drugs, toxins that appear in the body fluids. This accumulation has both positive and negative effects. For example, the body's lipids reserves retain both valuable and lipid-soluble vitamins such as A, D, E, K, and potentially dangerous lipid-soluble pesticides such as DDT. Steroids. Steroids are large lipid molecules that share a distinctive carbon framework. They differ in carbon chains They're attached to the basic structure. Steroid cholesterol, bile, and related steroids are important in the body. All animal cell membranes contain cholesterol. Cells need cholesterol to maintain their cell membranes, as well as cell growth and division. Steroid hormones are involved in regulation of sexual function. Examples include sex hormones such as estrogen and testosterone. Again, we will go into these types of hormones later in the lecture series. Steroid hormones are important in the regulation of tissue metabolism and mineral balance. Examples include hormones of the adrenal cortex called corticosteroids, and calcitrol, a hormone important in the regulation of calcium ion concentration in the body. Bile salts. Steroid derivatives called bile salts are required for the normal processing of dietary fats. Bile salts are produced in the liver and secreted in the bile. They interact with lipids in the intestinal tract and facilitate the digestion of absorption in lipids. Later in the lecture series, when we discuss the digestive tract, you will see the complete details of digestive substances such as bile and other enzymes involved in the digestive system. Cholesterol is obtained in two ways. One, by the absorption from animal products in the diet, and two, by synthesis within the body. Dietary cholesterol. Liver, fatty meats, cream, egg yolks are especially rich in dietary sources of cholesterol. A diet high in cholesterol can be harmful because a strong link exists between high blood cholesterol levels and heart disease. Current nutritional advice suggests limiting cholesterol intake to less than 300 milligrams per day. This amount represents 40% reduction for the average adult in the United States. Unfortunately, because the body can synthesize cholesterol as well, blood cholesterol levels can be difficult to control by dietary restriction alone. There is a connection between blood cholesterol levels and heart disease. Again, later in the lecture series, you will have a lecture concerning cholesterol and its relationship with heart disease. Phospholipids and glycolipids. Phospholipids and glycolipids are structurally related, and our cells can synthesize both types of lipids primarily from fatty acids. In a phospholipid, a phosphate group links the diglyceride and the non-lipid group together. In a glycolipid, a carbohydrate is attached to the diglyceride. Note that placing lipid last in the name indicates that the molecule consists of primarily lipids. This is an illustration taken from your textbook demonstrating phospholipids and glycolipids. Phospholipid lecithin, in a phospholipid, a phosphate group links a non-lipid molecule to a diglyceride. In a glycolipid, a carbohydrate is attached to the diglyceride. Micelles, in large numbers, phospholipids and glycolipids form a micelle. In the hydrophilic heads facing the water, molecules and the hydrophobic tail on the inside of each droplet. Hydrophobic and hydrophilic. Again, these are terms we have alluded to previously in the lecture, but a long hydrocarbon tail, phospholipids and glycolipids are hydrophobic, but the opposite ends of the non-lipid heads are referred to as hydrophilic. In water, large numbers of these molecules tend to form droplets, referred to as micelles, with the hydrophilic portion favoring the aqueous medium of water, whereas the hydrophobic end is pointed away from the aqueous environment or the water. Most meals contain a mixture of lipids and other organic molecules, and micelles form as food breaks down in the digestive tract. In addition, phospholipids and glycolipids, micelles may contain other insoluble lipids such as steroids, glycerides, and long-chain fatty acids. Structural lipids such as cholesterol, phospholipids, glycolipids are called structural lipids because they help form and maintain intracellular structure called membranes. At the cellular level, membranes are sheets of layers composed primarily of hydrophobic lipids. Cell membrane. A membrane surrounds each cell and separates the aqueous solution inside the cell from the aqueous solution in the extracellular environment. A variety of internal membranes subdivide the interior of the cell into specialized compartments, each with a distinctive chemical nature, as a result, different functions. Proteins are the most abundant organic component of the human body, and in many ways are the most important. The human body may contain as many as 2 million different proteins and can account for about 20% of the total body weight. All proteins contain carbon, hydrogen, oxygen, nitrogen, and a smaller quantity of sulfur may also be present. The functions of proteins are important to know. I will go over them at this time, but we will also go over the function of proteins later when we talk about the cell membrane. One of the functions of proteins includes support. Structural proteins create a three-dimensional framework for the body, providing strength, organization, support, cell structures, tissues, and organs. Movement. Another valuable function of proteins. Contractile proteins are responsible for muscular contractions related to proteins and responsible for the movement of individual cells. Transport. Insoluble lipids, respiratory gases, special minerals such as iron, several hormones cannot be transported in the blood unless they are first bound to special transport proteins. Other specialized proteins transport materials from one part of the cell to another. Another function includes buffering. Proteins provide a considerable buffering action and thereby help prevent dangerous changes in pH in our cells and other tissues. Metabolic regulation. Enzymes accelerate chemical reactions in cells. The sensitivity of enzymes to environmental factors is extremely important in controlling the pace and the direction of metabolic operations. Proteins also function in coordination and control. Protein hormones can influence the metabolic activity of every cell in the body or affect the function of specific organs or specific organ systems. Proteins also function in defense. A tough, waterproof protein of the skin, hair, nails protect the body from environmental hazards. Proteins called antibodies, components of the immune system response, help protect us from disease. There are also special clotting proteins that restrict bleeding after an injury to the cardiovascular system. The structure of proteins. Proteins consist of a long chain of organic molecules called amino acids. 20 types of amino acids occur in significant quantities in the body. A typical protein contains 1,000 amino acids. The largest protein complexes have 100,000 proteins or more. Each amino acid consists of central carbon atoms which have 4 groups attached. Amino acids. Each amino acid consists of a central carbon atom which has 4 different groups are attached. A hydrogen atom, an amino acid group, a carboxylic acid group, and a variable group designated by R. This is the structure of an amino acid. Again, this illustration is taken from your text. As you can see, the central carbon, located here, and then an amino group, or carboxylic acid group, and then a variable group related, again, it is illustrated by the R group. The name amino acid refers to the presence of an amino group and a carboxylic acid group, which all amino acids have in common. The different R group that distinguishes one amino acid from another, given each of its own chemical properties. All amino acids are relatively small, water-soluble molecules. There are 20 amino acids commonly found in proteins. In a normal pH range, the body fluids, the carboxylic acid groups on the main amino acid releases hydrogen ions. When this occurs, the carboxylic acid group changes from its typical pattern to, and an amino acid becomes negatively charged. Peptide bonds. Amino acid glycine and alanine link to form a dipeptide. Peptides form as dehydration synthesis creates a peptide bond between the carboxylic acid group of one amino acid and the amino acid group of another. Peptide bonds. As previous slides indicate, dehydration synthesis can link two amino acids. The process creates a covalent bond between carboxylic acid groups and one amino acid and the amino group of another. Such a bond is known as the peptide bond. Molecules consisting of amino acids held together by peptide bonds are called peptides. A molecule created in this example is called dipeptide because it contains two amino acids. Attaching a third amino acid produces a tripeptide next to a tetrapeptide, a pentapeptide, and so forth. Tripeptides and larger peptide chains are called polypeptides. Polypeptides containing more than 100 amino acids are usually called proteins. Proteins are negatively charged. You may be familiar with the names of several important proteins, including hemoglobin in the red blood cells, keratin in fingernails, and in hair. Again, these particular constituents will be discussed in a lot more detail later on in the lecture series. Because each protein contains amino acids, they are negatively charged. The entire protein has a net negative charge. The characteristics of particular proteins are determined in part by the R group on its component of the amino acid. But the properties of a protein are more than just the sum of properties in its part, for polypeptides can have a highly complex shape. Proteins have 4 levels of structural complexity, which will be discussed. They have a primary structure, which is a sequence of amino acids along the length of a single polypeptide. This illustration, taken from your text, gives a demonstration of the primary structure. A secondary structure results from bonds that develop between atoms at different parts of the polypeptide chain. Hydrogen bonding, for example, may create a simple spiral known as a alpha helix or a flat pleated sheet, which one form depends on the sequence of amino acids in the peptide chain and where hydrogen bonding occurs along the length of the peptide. The alpha helix is the most common form, but a single polypeptide chain may have both helical and pleated sections. Again, same illustration demonstrating to you the alpha helix, which is the most common form, but a single polypeptide chain may have both helical and pleated sections. Again, the alpha helix. Tertiary structure is a complex coiling and folding that gives the protein its final three-dimensional shape. Tertiary structures result primarily from interactions between polypeptide chains and the surrounding water molecules, and to a lesser extent from interactions between the R groups of amino acids in different parts of the molecule. The tertiary structure, most such interactions are relatively weak. One, however, is very strong. That's the disulfide bond, a covalent bond that may form between two molecules of amino acid, cysteine, located at different sites along the chain. We will revisit the disulfide bond later in the lecture series. The disulfide bond creates a permanent loop or coil in the polypeptide chain. Quaternary structures is an interaction between individual polypeptide chains to form a protein complex. Each of the polypeptide subunits has its own secondary and tertiary structures. Quaternary structure, protein hemoglobin, contains 4 globular subunits. Hemoglobin is found within red blood cells, where it binds and transports oxygen. In keratin and collagen, 3 alpha helical polypeptides are wound together like strands of rope. Keratin is a tough, water-resistant protein at the surface of the skin and in the nails and the hair. Proteins fall into 2 general structural classes on the basis of their overall shape and properties. These include fibrous and globular proteins. Fibrous proteins take the form extended sheets of strands. These shapes are usually the product of secondary structure, as in the case of proteins that exhibit the pleated and sheet configuration, or quaternary structures, as in the case of keratin and collagen. Fibrous proteins are tough, durable, and generally insoluble in the body and usually play structural roles. Globular proteins are compact and generally rounded, readily enter solution. A unique shape of each globular protein is the product of its tertiary structure. An example of globular protein. Myoglobin, a protein in muscle cells, is a globular protein, as is hemoglobin, the oxygen-carrying pigment in your red blood cells. Many enzymes, hormones, and other molecules that circulate in the bloodstream are globular proteins, as are the enzymes that control chemical reactions inside cells. These proteins may function only so long as they remain in solution. Among the most important of all body's proteins are enzymes. These molecules catalyze the reactions to sustain life. Almost everything that happens inside the human body does so as a specific enzyme makes it possible. Substrates. The reactants in an enzymatic reaction are referred to as substrates. As in other types of chemical reactions, the interaction among substrates yields specific products. An active site. Before an enzyme can function as a catalyst to accelerate a chemical reaction without itself being permanently changed or consumed, the substrate must bind to a specific region of the enzyme. This region, called the active site, is typically a groove or a pocket into which one or more of the substrates will nestle like a key fitting into a lock. The physical fit is reinforced by the weak electrical attractive forces such as hydrogen bonding. The tertiary or quaternary structures of the enzyme molecule determine its shape and its active site. Although enzymes are proteins, many organic or inorganic compounds that will bind to the active site will be a substrate. This illustration, again, is taken from your textbook, a simplified view of the enzyme structure and function. Each enzyme contains a specific active site somewhere on its surface, on its exposed surface. Again, as I mentioned earlier in this lecture, it's like the substrate fits into a key, fits into the slot. These are the enzymatic steps involved. Step 1, substrate binds to an enzyme at its active site. It appears likely that the substrate binding results in a temporary reversible change, the shape of a protein, that its changes furthers the reaction by placing physical stresses on the substrate molecule. Step 2 and 3. Step 2, the enzyme then promotes production and formation, And then in step 3, the completed product detaches from the active site and the enzyme is free to repeat the process. Enzymes work quickly, cycling rapidly between substrates and products. For example, an enzyme providing energy during a muscular contraction performs a reaction sequence 100 times per second. Enzymes that catalyze a synthesis reaction. All enzymes have 3 basic characteristics. They include specificity, saturation limits, and regulation. Specificity, as the term implies, each enzyme catalyzes only one type of reaction that can accommodate only one type of substrate molecule. The property is called specificity. The specificity of an enzyme is determined by the ability of a particular substrate to bind to the active site, which makes up a relatively small portion of the entire protein. Differences in an enzyme structure that do not affect the active site or change the response to enzymes to a substrate binding do not affect the function of the enzyme. Different tissues typically contain enzymes that differ slightly in structure but catalyze the same reaction. Such enzyme variants are referred to as isoenzymes. Saturation limits. The rate of an enzymatic reaction is directly proportional to the concentration of substrate, molecules, and enzymes. We alluded to this in lecture 2 of the lecture series. An enzyme molecule must encounter appropriate substrates before it can catalyze the reaction. The higher the substrate concentration, the more frequent the encounters will be. When substrate concentrations are high enough that every enzyme molecule is cycling through its reaction sequence at top speed, further increases in substrate concentration will not affect the rate of the reaction unless additional enzyme molecules are provided. The substrate concentration required to have a maximum rate of reaction is referred to as the saturation limit. This is a term, again, you should become familiar with. An enzyme that has reached its saturation limit is said to be saturated. To increase the reaction rate further, the cell must increase the number of enzyme molecules available. This is one important way that cells promote specific reactions. Regulation. A variety of factors can turn enzymes on or off and thereby control reaction rates inside the cell. The presence or absence of cofactors can control the rate of reactions inside cells. Anything that changes the tertiary or quaternary shape of an enzyme may turn it on or turn it off. Regulation. Each cell contains an assortment of enzymes, and any particular enzyme may be active under one set of conditions and inactive under another set of conditions. Because the change is immediate, enzyme activation or inactivation is an important method of short-term control over reaction rates and pathways. Cofactors and enzyme function. A cofactor is an ion or a molecule that must bind to the enzyme before substrates can also bind. Cofactor is a term that you should become familiar with. Without a cofactor, the enzyme is intact, but non-functional. With the cofactor, the enzyme catalyzes a specific reaction. Examples of cofactors include ions such as calcium, magnesium that bind the enzyme at its active site. Cofactors may also bind to other sites as long as they produce a change in the shape of the active site that makes the substrate binding possible. Coenzymes are non-protein organic molecules that function as cofactors. Coenzymes is another term that you should become familiar with. Our bodies convert many vitamins into essential coenzymes. Vitamins are structurally related to lipids or carbohydrates, but have unique functional roles. The human body cannot synthesize most vitamins it needs, so it must be obtained from the diet. Some other important factors in enzyme function. Temperature and pH. We have discussed pH earlier in the lecture series. Each enzyme works best at a specific temperature and a pH value. For the human body, if you remember, the normal pH value is 7.35 to 7.45. Again, those are numbers that you should have memorized at this point. As temperature rises, protein shape changes and enzyme function deteriorates. Death occurs at very high body temperatures above, and because proteins undergo denaturization, then a change in the tertiary and quaternary structures will occur. Denaturation. Because denatured proteins are non-functional, the loss of structural proteins and enzymes should cause irreparable damage to organs and organ systems. Denaturation can be temporary or permanent. Eventually, the proteins become completely and irreversibly denatured. Enzymes are equally sensitive to pH changes, changes in pH. Pepsin, an enzyme that breaks down proteins and the contents in the stomach, work best at a pH of 2.0, which is strongly acidic. Your small intestines contains trypsin, another enzyme that attacks proteins. Trypsin works only in an alkaline environment with an optimum pH of 7.7, which is weakly basic. Glycoproteins and proteoglycans. Glycoproteins and proteoglycans are combinations of proteins and carbohydrate molecules. Glycoproteins are large proteins with small carbohydrate groups attached. These molecules may function as enzymes, antibodies, hormones, protein components, and cell membranes. Glycoproteins and cell membranes play a major role in the identification of normal versus abnormal cells, as well as the initiation, coordination of the immune response. This will be discussed in more detail later in the lecture series. Glycoprotein secretions called mucins absorb water and mucus. Mucus coats the surfaces of the respiratory tract and the digestive tract, providing lubrication. Proteoglycans are large polysaccharide molecules linked by polypeptide chains. Proteoglycans in tissue fluids give them the syrupy consistency. Nucleic acids. Nucleic acids are large organic molecules composed of carbon, hydrogen, oxygen, nitrogen, and phosphorus. Nucleic acids store the process information at the molecular level inside the cell. Two classes of nucleic acids: deoxyribose nucleic acid, or DNA, a term you may already be familiar with, and ribonucleic acid, or RNA, again another term you may already be familiar with. DNA in our cells determines our inherited characteristics such as our eye color, hair color, and blood type. DNA affects all aspects of the body's structure and function because DNA molecules encode the information needed to build proteins. By directing the synthesis of structural proteins, DNA controls the shape and physical characteristics of our body. By controlling the manufacture of enzymes, DNA regulates not only protein synthesis, but all aspects of cellular metabolism, including the creation and destruction of lipids, carbohydrates, and other vital molecules. Several forms of RNA cooperate with the manufacture of specific proteins by using information provided in DNA. Nucleic acid consists of nucleotides linked by dehydration synthesis. Each nucleotide has three components. These three components include a sugar, a phosphate group, and a nitrogenous base. The sugar is a pentose sugar, a 5-carbon sugar, either ribose in RNA or deoxyribose in DNA. Each pentose is attached to a phosphate group and to a nitrogenous base. 5 nitrogenous bases occur in nucleic acid: adenine, guanine, cytosine, thymine, and uracil. Purines and pyrimidines both are nitrogenous bases of nucleic acid. the illustration here gives you the representation and the chemical structure for these various purines and pyrimidines. Adenine and guanine are double-ring molecules called purines. The other three bases, cysteine, uracil, and thymine, are single-ring molecules referred as pyrimidines. Both RNA and DNA contain adenine, guanine, cysteine. Uracil, important to note, is only in RNA, and thymine, also important to note, is only in DNA. The formation of nucleic acid. In the formation of nucleic acid, a nitrogenous base is attached to a pentose molecule. A nucleotide forms when a phosphate group binds to the sugar. Dehydration synthesis attaches the phosphate group to one of the nucleotides and carbohydrates of another. The backbone of nucleic acid molecule is a linear sugar to phosphate to sugar sequence with nitrogenous bases projecting to one side. The role of nucleic acid is the storage and transfer of information, specifically information essential to the synthesis of proteins within our cells. Regardless of whether we are speaking of DNA or RNA, it is the sequence of nitrogenous bases that carries the information. Important structural differences distinguish RNA from DNA. A molecule of RNA consists of a single chain of nucleotides. Its shape depends on the order of the nucleotide and the interaction among them. Nucleic acids are long chains of nucleotides. Each molecule starts at a sugar of the first nucleotide and ends at a phosphate group in the last member of the chain. Again, this illustration is from your textbook, demonstrating an RNA molecule which has a single nucleotide chain. RNA molecule shape is determined by the sequence of nucleotides and their interactions among them. A DNA molecule has a pair of nucleotide chains linked by hydrogen bonding between complementary base pairs. Three types of RNA exist. Human cells have three types of RNA. These include 1. Messenger RNA, 2. Transfer RNA, and 3. Ribosomal RNA. These types have different shapes and functions, but all three are required for the synthesis of proteins. DNA molecules consist of a pair of nucleotides chains. Hydrogen bonding between opposing nitrogenous bases holds the two strands together. The shapes of the nitrogenous bases allow adenine to bind only to thymine and cysteine, for example, to bond only to guanine. In complementary base pairs, as a result, combination of adenine-thymine and cysteine-guanine are known as complementary base pairs. And the two nucleotide chains in DNA molecule are known as complementary strands. Cells use one of the two complementary DNA strands to provide information to form a single strand of messenger RNA. The instructions needed to synthesize specific proteins are spelled out in the sequence of nitrogenous bases along the messenger RNA strand. Ribosomal RNA reads those instructions and, with the help of transfer RNA, assembles amino acids in the proper sequence to create the desired protein. Two strands of DNA twist around one another in a double helix, resembles a spiral staircase. Each step of the staircase corresponds with one complementary base pair. This is a table from your textbook, Table 2.6, which compares RNA with DNA. You should be familiar with the items on this table, basically giving you the sugars that are involved, ribose for RNA, deoxyribose for DNA. The nitrogenous bases is listed in both, paying close attention to uracil, being only located in RNA, and thymine, which is only in DNA. And then the number of nucleotides, the shape of the molecule, and the function, which you should review. Now we'll talk about some high-energy compounds, also important in the human system. To perform their various vital functions, cells must use energy obtained by breaking down organic substances, a term referred to as catabolism, which again we discussed in Lecture 2. To be useful, that high energy must be transferred from one molecule to a molecule from one part of the cell to another. The usual method of energy transfer involves the creation of high-energy bonds by enzymes within our cells. A high-energy compound is a covalent bond whose breakdown releases energy in the cell, and this energy can be harnessed. In human cells, high-energy bonds generally connect to phosphate groups to organic molecules. The resulting complex is a high-energy compound. The attachment of phosphate groups to another molecule is referred to as phosphorylation. This process does not necessarily produce high-energy bonds. The creation of a high-energy compound requires a phosphate group, an enzyme capable of catalyzing a reaction, and a suitable organic substrate which the phosphate group can be added. The most important of such substrates is adenosine, a combination of adenine and ribose with two phosphate groups attached. This compound, adenosine diphosphate, or ADP, is created by the phosphorylation of the nucleotide adenosine monophosphate, AMP, a building block in nucleic acids. A significant energy input is required to convert AMP to ADP, and a second phosphate is attached to the high-energy bond. Even more energy is required to add a third phosphate group, thereby producing a high-energy compound referred to as ATP. A molecule of ATP consists of adenosine, adenine plus ribose, to which three phosphate groups have been joined. Cells most often transfer energy by attaching a third phosphate group to ADP, a high-energy bond, and then removing that phosphate group at another site where the associated release of energy performs cellular work. The illustration here typifies that by seeing adenine combining with ribose and phosphate, and you see recurrent phosphorylations occur leading to ATP. The conversion of ADP to ATP and the reconversion of ATP to ADP is the most common method of energy transfer in our cells. Conversion of ATP to ADP requires an enzyme. These proteins we've discussed before. Adenosine triphosphatase, ATPase, is another name used for that. Throughout life, our cells continuously generate ATP from ADP and use the energy provided by ATP to perform vital functions such as synthesis of protein and contraction of muscles. Although ATP is the most abundant high-energy compound, there are others, typically other nucleotides, that have undergone phosphorylation. The example, guanosine triphosphate, GTP, and uridine triphosphate, UTP, and nucleotide-based high-energy compounds that transfer energy to specific enzymatic reactions.